This is what I would argue, one of, if not the most important concept that you should have learned in an introductory to biology course before you get to anatomy. And that is, how do things get across the plasma membrane? How do things diffuse? How do things move? This is really essential, especially when we're starting to talk about things like excitable membranes. So membranes that change their voltage in response to ion flow. But this is also going to be really important when we think about concentrations in digestion and other areas. So I wanted to just briefly show us, right, when we've got our plasma membrane, right? So I'm going to draw a couple of different things here. We've got our plasma membrane with its phospholipid bilayer. Right, and a reminder, the phospholipid bilayer is primarily hydrophobic because of these tails. And these tails being hydrophobic, right, so they really only want hydrophobic things around them. So if this is a phobic region, we want hydrophobic particles to be able to get through. Phobic. Now these phobic particles, they can get through if they're small enough, right? So if they're small, they can just go through the membrane. And we refer to that as diffusion. Okay? And it can diffuse through the membrane only in certain conditions. Okay? So a lot of times when I'm talking to students, they'll say, well, it's a hydrophobic, so it's going to get through. But, right, one, it needs to be small enough, but also we need to have what's referred to as a chemical gradient. Right? We need a chemical gradient. What that means is that there's a chemical, right, so this phobic molecule, and that there is a gradient, meaning that there is more on one side than the other side, right? So let's say we have tons and tons and tons of that phobic molecule on the outside, right? So that phobic molecule up there, and there's maybe only like one on the inside. Because it's so crowded, right? If you're at a crowded party, you don't want to be in the crowded room. You want to be able to breathe and move and do your thing. And so more of those particles are going to be able to move in to the cell. But I want to point out, right, this is something that we also tend to forget, is that eventually there are as many of these phobic particles on the inside as there were on the outside. And so once we have an equal amount between them, we have what's referred to as an equilibrium. Our cells sometimes want an equilibrium, sometimes they don't. And it really depends on what processes are taking place and what is being used, okay? But once we hit equilibrium, the movement kind of stops going in that one direction and can now go in both directions, okay? So we reach that equilibrium, and so now you could go in or out based on diffusion, okay? The other thing that will move by diffusion is going to be things like gases. So when you get to the respiratory system, you will need to consider, how do carbon dioxide and oxygen diffuse through that membrane, and when is it going to want to go into the cell versus when does it want to go out of the cell? It's going to follow that chemical gradient unless our cell does something to manage it. So let's talk about the cells that are too big, right? So what about a cell that doesn't have, let's talk about the molecules that are too big, right? So let's say you've got a nice hydrophobic molecule, but it can't get through. What it's going to usually go through is a protein within the cell membrane to be able to go down through. Sometimes they're carried through a carrier protein. Sometimes it's through a channel. And it really depends on the molecule. These proteins, I want to point out, we talked about gene expression. These particular proteins on the plasma membrane are not always there, and their concentration is not always the same. It's what our body needs. A good example of this would be glucose, which is regulated by insulin. And so sometimes we have a lot of glucose transporters, and sometimes we don't. And that's dependent on insulin. So these proteins are highly regulated to make sure these big molecules move in. I want to point out that these carriers can function in two ways, right? So I've got these these larger proteins can function kind of in two different ways. The first way can require no energy, and we refer to that as facilitated diffusion. When it does facilitated diffusion, right, so no energy, no ATP, just like normal diffusion, we're going down our chemical gradient. We're going from a high concentration to a low concentration. But our body doesn't always want to do that, actually. Our body actually sometimes really needs to pump something out. Like, think of acids in our stomach, right? My stomach's already got a ton of acid, but I need to keep making acid so I can keep breaking down whatever food gets into my stomach. So sometimes we're going to use energy in the form of ATP to do something that we don't already want to do, right? So think about, I don't want to get out of bed in the morning sometimes, but if I give myself a little oomph, I'll get myself out of bed and do what I need to do. Same with the cell. That protein may not want to do the action, but with ATP and a little oomph, it'll be able to do that reaction. When it does this, we call this active transport. And so it's going up its chemical gradient usually. And it's usually going from a low concentration to a high concentration. There is actually two forms of that active transport. So the normal one, where we're just using one protein and just going up that gradient, is called primary active transport. There is another one where we use two different proteins. We refer to that as secondary active transport. And this is sort of what secondary transport looks like. So I've got my cell, okay, and we've got two different proteins. Now, sometimes they're on the same side of the cell, sometimes they're on opposite sides of the cell. I'll just separate them to make it easier. Let's say there's a molecule that we want to go in. Let's say it's an ion that we want to go in. This ion wants to go into the cell, but it's not going to be able to get in there unless it has that chemical gradient. What we can do is right, so let's say we already have an equilibrium inside, and we want to make it so that there is a gradient. What we can do is, right, let's say there's a ton on the other side here. If I want to get this ion out so we can have a high to low, I can take these through this protein. But look, there's only 5 here, and there's more than 5 outside, right? So it's going from a low concentration to a high concentration. Down here is active transport, where we're going to use some ATP to get these ions out. Now that the ions are out, we have 5 out here and 2 in here. Now we have a high concentration to low, and this is going to be that facilitated diffusion. So this is why it's called secondary active transport, where we do have an active transport, but it's for a secondary function. I hope this is helpful for you guys as you're going through this material.